Glycolysis in 10 steps. Glycolysis, the metabolic process serves as the foundation for both aerobic and anaerobic cellular respiration. In glycolysis, glucose is converted into pyruvate. Glucose is a six-membered ring molecule found in the blood and is usually a result of the breakdown of carbohydrates into sugars. It enters cells through specific transporter proteins that move it from outside the cell into the cell cytosol. All of the glycolytic enzymes are found in the cytosol. In step 1, one glucose molecule is converted to glucose 6-phosphate which catalyzed by hexokinase, and ATP is used up. In step 2, glucose 6-phosphate rearranged to become fructose 6-phosphate which catalyzed by the isomerase. In step 3, fructose 6-phosphate is added with another phosphate group from hydrolysis of ATP to form fructose 1,6-biphosphate. The second ATP is used. In step 4, fructose 1,6-biphosphate produced is split into glyceraldehyde 3-phosphate GAP and its isomer dihydroxyacetone DHAP, which are both catalyzed by aldolase. 5. DHAP rearranged into another molecule of GAP which catalyzed by phosphotriose isomerase. In step 6, GAP is oxidized. Hydrogen atoms are removed and NAD plus is reduced to become NADH. An inorganic phosphate is attached to the substrate to produce glycerate 1,3-biphosphate. Step 7. A phosphate from glycerate 1,3-biphosphate is transferred to ADP to form ATP. Glycerate 3-phosphate is formed. At glycerate 3-phosphate rearranged to form glycerate 2-phosphate which catalyzed by the phosphoglycerate mutase. In step 9, water is removed from the glycerate 2-phosphate to produce phosphophenol pyruvate, which are catalyzed by enolase. In step 10, phosphophenol pyruvate is converted to pyruvate, which catalyzed by pyruvate kinase. At the same time, another phosphate is transformed to ADP to form ATP. In step 1 and step 3, 2 ATP is used up, and in step 7 and step 10, 4 ATP is produced. Therefore, the net ATP produced is 2.
So Krebs cycle is a process oxidizes glucose derivative, fatty acids, and amino acids to carbon dioxide through a series of enzyme control steps. The purpose of Krebs cycle is to collect 8 high-energy electrons from these fields by oxidizing them, which are transported by activated carrier NADH and FADH2 to the electron transport chain. Here's an overall equation of the whole cycle. So in the first reaction is the formation of citrate, whereby acetyl-CoA combines with oxaloacetate, the first compound, in a condensation reaction to release citrate, or in other words, citric acid, catalyzed by citrate synthase. A coenzyme, CoA, is released and then followed by the formation of isocitrate. Citrate rearranged by the removal of water molecules, followed by the addition of water molecules to a different carbon atoms to form its isomer, isocitrate. Then is the oxidation of isocitrate to alpha-ketoglutarate. So firstly, isocitrate is oxidized to form oxalosuccinate first, and then at the same time, NADH plus is reduced to become NADH. Then the unstable oxalosuccinate bounded to isocitrate dehydrogenase is immediately decarboxylated, losing the carbon dioxide molecules and converts to alpha ketoglutarate. Then reaction 4, whereby it's oxidation of alpha ketoglutarate to succinyl CoA. Takes place here, producing succinyl CoA and a CO2 and coenzyme A and NADH is generated here, catalyzed by alpha ketoglutarate dehydrogenase complex. Then next is conversion of succinyl CoA to succinate. Substrate level phosphorylation occurs here. Succinyl CoA is converted to succinate. The energy release is used for the phosphorylation of GDP to GTP. Then GTP transfer a phosphate group to ADP to form ATP. Moving on is the oxidation of succinate to fumarate. Succinate is oxidized to form fumarate, and 2 hydrogen atoms are transferred to FAD to form FADH2. Then is the hydration of fumarate to malate. Fumarate becomes hydrated by an addition of water, then converts to malate, catalyzed by fumarase. And finally is the oxidation of malate to oxaloacetate, whereby malate is oxidized to regenerate oxaloacetate, and the NADH plus is reduced to become NADH. Thus, the cycle can be repeated again using oxaloacetate from the first reaction. The overall ATP generation is 12 ATP. Then moving on is the hardest electron transport chain. This pathway is the most efficient method of producing the initial substrate for this. The initial substrate for this cycle are the end products obtained from other cycles, namely Krebs cycles and glycolysis. And firstly, NADH acts as the first electron donor and get oxidized to NAD plus by complex 1, accompanied by the release of a proton out of the matrix. The electron is then transported to complex 2, which brings about the conversion of succinate to fumarate. Molecular oxygen acts as an electron acceptor in complex 4 and gets converted to water. Each enzyme complex carries out the transport of electron accompanied by the release of proton in the intermembranous space. Then the accumulation of proton outside the membrane gives rise to a proton gradient here. And this high concentration of protons, with all these protons here gathered, starts the process of chemiosmosis that activates ATP synthase complex, which is complex 5. Chemiosmosis refers to the generation of an electrical as well as a pH potential across a membrane due to the large difference in proton concentration. The ATP synthase utilizes this potential and acts as a proton pump to restore the concentration balance. For pumping the protons down here, the proton back into the matrix of the mitochondria, it also conducts the phosphorylation of ADP to ATP. So next in terms of redox reaction, the reduced coenzyme NADH, which is complex 1 here, functions to reduce the coenzyme Q10. This reaction donates electrons which are transferred through this complex using FMN and a series of ferrum sulfur clusters. The transport of this electron brings about the transfer of proton across the membrane into the intermembranous space. Then at complex 2, which acts on the succinate produced by citric acid cycle, which is Krebs cycle, converts it to fumarate, succinate to fumarate. This reaction is driven by a reduction and oxidation of FAD to FADH2, and along with the help of a series of ferrum sulfate clusters. This reaction also drives the redox reaction of quinone. These sets of reactions helps in the transporting of the electron to the third enzyme complex, which is complex 3. This complex oxidizes ubiquinone and also reduces 2 molecules of cytochrome C. The electron is transported via the reaction onto complex 4, accompanied by a release of protons. At complex 4, the receive of electron just now by a molecular oxygen here, to yield a water molecule. This conversion occurs in the presence of copper ions and drives the oxidation of reduced cytochrome C. The protons are pumped out during the course of this action, and then ATP synthase. The proton produced from the initial oxidation of NADH molecules and their presence in the intermembranous space give rise to a potential gradient, which causes chemiosmosis just now, and protons are transported back into the matrix, and ADP is converted to ATP. That's all from us. Thank you, and hope you understand Krebs cycle, glycolysis, and electron transport chain.